Hello, everyone. This is Xiaoxiao Wang from Nona Bioscience, and I'm so glad to have the opportunity to share our experience to use the Harbour Mice and the Beacon system for the therapeutic antibody discovery. Before I get into our main topics today, I would like to take a little bit of time to introduce Nona Bio to all you guys. Nona Bio is a quite young company that actually less than 2 years ago, we were still called Harbour Biomed, a biopharmaceutical company established in 2016 and listed in the Stock Exchange of Hong Kong at the end of 2020. 2 years ago, the company was just split into 2 subsidiaries based on different business models. And the Harbour Therapeutics showed here will focus on the R&D enabling and the clinical development of portfolio projects, especially the R-related projects, and further promote the international cooperations of these projects. And Nona Bioscience has inherited the full technology platform of Harbour Biomedics and then further expanded it to new fields, and in the meantime provide service for discovery and development of various novel bios. And as shown here, the name of Nona is actually coming from one of the 3 goddesses of fate from Roman mythology. She starts the stress of the life and enable others to succeed. Just as our name indicated, Nona Bioscience is committed to provide integrated discovery solution for biotech and biopharmaceutical companies from idea to R&D, and with our industrial leading platforms and experienced team for antibody discovery and development. One of the core technology platform of Nona is just as shown here, the Harbour Mice. It's one of the few full human transgenic mice platform with full IP protection and clinical validations, and now still can be accessed by public. To date, Harbour Mice have been recognized by many global partners and has been applied in more than 200 antibody discovery projects, of which more than 18 projects have entered the clinical stage. And another core technology is just shown here is the high throughput screening platform with Beacon single B-cell system, which will help us to get panels of lead antibodies from our Harbour Mice with great diversities and properties just in a single day. And as the first company start to use Beacon system in China, Nona has full experience with this system. Today, more than 80% of antibody generation projects are based on this technology in our company, especially the candidate antibodies in our deal with Pfizer and AZ are coming from Beacon technology. Well, I think many of you already get to know the power of Beacon system from different resources or even your own experience. So today, I won't take too much time to emphasize again why this technology are so powerful and so important for the therapeutic antibody discovery, but would like to share some of our own experience or tricks about how can we take the best advantage of Beacon system with our Harbour Mice to get great antibodies. As shown here that to get the lead antibodies with Beacon system, there are 3 major steps. First, to enrich the antibody secreting cells from mice, and then to get positive clones with sequential in-chip assays, and finally to obtain sequence information by single-cell sequencing. So let's start our story just step by step. So first, how can we enrich the right cells for antibody secreting that could be used for the in-chip assay? As shown here, that after being activated by antigens, there are 2 major groups of antibody secreting cells can be generated. And one is the plasmablast that can be proliferated but with lower antibody secreting rate and short half-life, which located in both bone marrow and spleens. And the other is the mature plasma cells with much stronger antibody secretion activities and a long life, and mainly located in the bone marrow. So based on this theory, since majority of these mature plasma cells with high IgG secreting are located in the bone marrow, cells from bone marrow should show better behavior during single B cell screening compared with cells from spleen. But actually, during our experiments with our Harbour mice, as well as the wild-type mice shown in this page, we actually get some different results. So as shown here in the table, the positive numbers from the bone marrow, the positive numbers from bone marrow or the spleen-enriched antibody secretion cells from 9 independent experiments with both Harbour mice, as shown here, these two kinds of Harbour mice, as well as this wild-type mice listed here. And we can find that except 2 experiments from these wild-type mice that show a higher positive numbers of positive cells, the cells with from the bone marrow derived cells. Others are all show opposite results, no matter based on this antibody binding activity against the protein or the cell antigens. And these results just indicate that, and as shown here, is these results indicate that in our future SBC, only cells from spleen or lymph node should be enough for the SBC screening, and no bother to take time getting cells from bone marrow again. Actually, the answer is no. We know that besides the positive numbers we could get from SBC, the sequence diversity we could achieve, and also the later ELISA positive rate for the recombinant antibodies is also very important for our antibody discovery project. So here, we actually also evaluated the ELISA positive rate of the sequences that we can obtain from either bone marrows or spleens, and also analyze the sequences to see the diversity of the sequences from these 2 different organs. And here just shows 2 examples that just coming from the previous tables. This one is using the BsAb mice, and we can see that with the higher positive numbers from the bone marrow compared with the spleen and the lymph node, the ELISA positive rate is actually comparable. And in the meantime, we can see that from this clustering results based on the CDR sequences, the diversity, we can see improved diversity if we add in this bone marrow derived cells into the whole sequences we obtained. And here the other case is using the SBC results with our Harbour mice. And we can see that even the bone marrow derived cells just yield less positive clones compared with the spleen organs. But if we see the diversities, we can see that we still can identify some unique cluster of CDR sequences from bone marrow derived cells. So both these 2 results here just tell us that if we can include in bone marrow during SBC, it will bring us some benefit to the whole antibody discovery project, especially with the increased diversity of the sequences that we can obtain from SBC screening. Well, after we get cells from bone marrow and spleen of mice, we need to enrich the antibody secretion cells for the in-chip screening. And as shown here, during this step, we actually can find some interesting things that the 2 commercial kits that can help us to enrich the cells with different vendors are actually can enrich cells including the different subgroups. The Metani kit, which including one more step for the negative screening, will exclude CD45R positive plasmablast, whereas the stem cell kit would keep both parts of these cells. So what will happen if we screening using these 2 different subgroups of cells in the chip? And here just shows the results. We can see that if we checking for the positive rate for the CD45 negative and CD138 positive purities, the stem cell kit will yield actually only, I think it's around maybe 30 to 40% of cells show the 45R positive, which indicate the plasmablast. And if we comparing the positive clones from the cells that coming with different kits or the subgroups, you can see that the pure plasma cells coming from the Metanic kit show significantly higher positive clones, as well as the qualities that show a better PTM-free sequences, and also it indicates much higher diversities, that there has 8 indicated other unique CDR clusters comparing with the one coming from the both plasma and plasmablast. And here just the sequence analysis results, which just supported the conclusions that we have in the previous slide. And we can see that based on these CDR clusters and germlines, if we just excluded the CD45R positive portion of the cells, there should be higher diversity or more unique clusterings, or the germlines could be obtained. So here is just a summary for the antibody secretion cell enrichment, that we suggested to include cells from both bone marrow and spleen to help to get positive clones with improved diversity. And in the meantime, the plasma cells with higher purity will be preferred against a mixture of both plasma cell and plasmablast for a more efficient screening that can help us to get more positive clones and a better diversity of the sequences. And after we success enrich the cells for in-chip assay, many scientists may face such problem as showed here, that we may got too many cells, especially if we sacrifice more than one mice for one SBC. That there were too many cells and more than we can use for just one SBC screening with Beacon. So what we can do with the rest of the plasma cells? Can we use them in more batches of SBCs? And will such approach help us to increase the diversity or coverage of the sequences? To answer the questions, Nona first tried to see whether we can utilize these frozen plasma cells for another run of SBCs. And as shown here, the several experiments with different batches of frozen plasma cells were done to check the recovery rate of both IgG secretions as well as the antigen positive clones, compared with the same batch of the fresh plasma cells. And as shown here, that we can see after frozen store for days to months, we definitely can still use these plasma cells for single B cell screening, but the frozen storage of these plasma cells would cause the loss of IgG secreting activity, and thus decrease the number of positive clones that we can obtain from one SBC. And also, the frozen stocks for more than 2 months were not really suggested to use due to this, as we show like in the last 2 lines, that a dramatically decrease of the activity could be observed. And we also analyzed the sequences from SBCs with frozen cells and comparing it with the same batch of the fresh cell SBC. And we can see that the CDR-based clustering results show that with additional screening of these frozen stocks from the same batch of plasma cells can actually merely help to increase the diversity of the whole screening with this same batch of the plasma cells. Another series of experiments with cultured plasma cells were also performed to see how we can use in this extra of the plasma cells. And we can see here in the table that unlike using the frozen cells, after overnight culturing, the plasma cells actually could show a comparable activity as shown here with this H2R2 Harbour mice or the BsAb wild-type mice, or sometimes even the increased screening efficiency as shown here with the HCAB Harbour mice. But one thing needs to be noticed here that if we see the SBC 21-1 or SBC 21-4, these two assays, we can see that the positive rate will drop dramatically with cultured plasma cells if we perform multiple rounds of sequential assays. And we can see that for the fresh cells, the correlations or the positive rate keep comparable from the first in-chip assay to the fourth in-chip assay. But with the cultured plasma cell, it dropped dramatically. And also the sequence analysis was also done, and as what we did with the frozen plasma cell SBC. And as the results here just indicate that although the screening efficiency are much better for these overnight cultured cells compared with frozen cells, we still cannot get any significant benefits from the additional screening with these cells, because we can see that the diversity is not that good with these cultured plasma cells, and actually we cannot really getting improved diversities or coverage with additional screening of this SBC. So in conclusions, the frozen plasma cells are not really recommended for the SBC because it's decreased efficiency. And although the cultured plasma cells could maintain a good quality of SBC, we still don't suggest it to do these multiple rounds of SBC with same batch of enriched plasma cells, because not much improvement could be achieved with this sequence coverage or diversity with such approach. And in another word, actually our data here just indicate that the current throughput of one round of SBC with 1 to 2 chips that can screening around 10 to 20K cells with each SBC is actually high enough to cover the majority of positive hints with plasma cells from 1 to 3 mice. So next, we can start our in-chip assays. And we know that a lot of assay optimizations to achieve better results were done by the scientists from different labs, as well as the Bruker Cellular. So here I won't share any more assay optimization results, but just to talk about something different. And we know that one of the major advantages of Beacon compared to other single-cell screening or high-throughput screening technology is that we can do sequential in-chip assays, several sequential in-chip assays with one SBC, and help us to get these positive clones with multiple characters. But we also are worried about that. When the plasma cell, will the plasma cell lose activity after sitting in the small pans of the chip for a longer time? And this loss of the activity, will this cause the unreliable results for these sequential assays? So as shown here, we run the same binding assay sequentially in chip and check the change of positive numbers from round to round. The results here indicated that in general, the results could keep acceptable consistency from round to round up to 4 to 5 sequential assays. As shown here, the positive across the multiple assays still keep to around 70%, even with the 5th rounds. And also, if we check the overlap between two adjacent assays, we can see that these recoveries or overlap rate is higher than 90%, which is quite qualified to support the screening strategies with multiple criteria. And the same experiments was applied with overnight cultured plasma cells. And to be surprisingly, we just found that when we can see very good results for these sequential in-chip assays with fresh plasma cells, the sequential assay may not be suitable with these cultured plasma cells for this dramatically decreased function of assay that even just start with the second round of the binding assay, the positive rate cross multiple assays are dropped just under 50%. So if people really want to do SBC with cultured plasma cells, the sequential assay may not be suitable. And we know that with these fresh plasma cells, up to 4 to 5 rounds of sequential assay are applicable. But in the meantime, we may still see some inconsistency between each rounds. So in this case, people may also think about, can we use a multiplex assay instead of this sequential assay? Will it be a better choice? Will it have a better correlations? So as shown here that the multiple assay, not like this classic binding assay in chip assays, it's just using the antigen anti-IgG beads to catch the secretion antibodies to allow it to test the multiple antigen bindings in just one assay. Not like the traditional assays, it can only check the binding activity against the protein antigens, and in the meantime, the interaction between the antibody and the antigen is monovalent. So here just the results that we're comparing the binding results against the same antigen, but obtained with classic antigen beads assay and the multiplex assays. We can see that the multiplex assay could definitely detect the binding to different antigens in the same round, and the results is quite correlated to each other. But in the meantime, it shows much weaker signals and much lower sensitivities to get positive clones that we can see only, less than half of the positive clones could be get from the multiplex assay. And it's mainly due to its monovalent interactions, as well as the competitions among the different antigens in the channel in one assay. So in the conclusion, we can see that although these multiplex assays can help us to run or to detect multiple bindings in one assay, it's not really applicable for most of the situation due to its lower sensitivity. So if there is less binders we can achieve, and every binders are really valuable, we need to get out most of the binder out, we suggest to use these classic sequential assays. But if there are too many hits, and we just want to pick up ones with potential higher binding activity and better cross activities, then this multiplex assays can be used. So after that, we go to the single B-cell sequencing steps. And some of you may already notice from previous data that when using one of our Harbour mice, the HCAP mice, which is a full human, heavy chain only mice, we can always getting a lot of clones or much more clones than other strain of mice. So in this situation, our scientists just developed this multi-cell sequencing method with NGS, allowing us to sequence more cells in one well and keep the comparable sequence recovery rate as the single cell sequencing as shown in this table. And also in the meantime, if we're checking for the diversity of the sequences, we can see that by using this method, we could get more sequences with increased diversity comparing with the traditional single B-cell sequences. But since this method can only be applied with our HCAP mice, which not require the pairing of heavy chain and light chain because these mice can only produce antibodies with only heavy chains, I won't go details here in this talk. So finally, here are just some case studies to show how we can use the beacons and to do these antibody discovery projects. First, here shows a case study that used the beacon to identify antibodies, recognize these multi transmembrane antigens, the Claudin 18.2. So we can see that with the sequential assay of the cell-based assay for both positive and negative screening, we can finally identify more than 200 unique sequences. And the later recombinant antibody screening or evaluation indicated that all of these antibodies show pretty good binding to both human and cyno antigens, and in the meantime, the minimum binding to the paralog, the Claudin 18.1. And also the final leads that identified from this approach also show pretty good biophysical properties. And here shows the comparison between the SBC and hybridoma, the traditional screening. And we can see that in this project, we use both SBC and hybridoma for the screening. By using SBC, we can screen definitely more cells, the 40,000 of cells, just within 3 weeks, and to obtain 44 unique sequences finally, comparing with only 14 sequences from hybridoma in 3 months. So we know that by using SBCs, we can get in much better diversity sequences in a less time. And also people may also curious about how the comparison between SBC and other high throughput screening, especially the NGS. Here just to show one case study that comparing the sequences results between the SBC and NGS. We can see that though SBC could retrieve less sequences than NGS-based screenings, its sequences could cover majority of the diversity from NGS. And in the meantime, if we checking for this red branches, we can see that SBC could also yield some unique clusters that could not be identified by NGS. And if we add in the AI-based screening with NGS together, what will happen? So here, in this case studies, we need to get the candidate antibodies, show the cross-reactivity for both human and cyno targets, but in the meantime, no binding to its paralogs. So we can see that after doing the NGS and AI-based screening, we can get quite similar numbers of human and cyno cross-active positive clones. But all of these clones will show the binding to one or even more paralogs, comparing with the SBC that we can get in no binders with these paralogs because counter-screening, sequential counter-screening chip screening is approached. So in this case, we know that the beacon-based SBCs will show advantages to gather leads with more complex conditions. And here just coming the taking home messages of my talk today. With the optimized process of antibody secretion, cell enrichment, in chip assays, and single-cell sequencing, Beacon will help scientists to obtain the therapeutic antibodies in a high-throughput and high-efficient manner that hundreds of leads with desired properties and diversity could be identified in weeks, compared with only a few sequences with low diversity from traditional hybridoma in months. And if we're comparing the SBC with other high-throughput screening method, especially the one based on NGS and AI, Beacon will help to identify some unique sequences that may be lost during the biopsy sample preparation process in the NGS. And it will help to identify leads to meet multiple binding requirements with its sequential in-chip assays. And thank you for all your attending for my talk. And if we have more questions, we can discuss in the later discussion panel.